So CMV is a virus, it's a DNA virus. It's a virus that infects most people in the world. It's a virus that hides. Most of us don't know we've got it. And most of us go through life without it ever causing a problem.
If a woman during pregnancy is infected with CMV, they can transmit it to the child in the uterus, and that results in congenital CMV infection.
Every year there are about 20,000 children in the US born with CMV infection. Of them, about 6,000 to 7,000 they'll end up with deafness, with blindness, with brain damage, with microcephaly, with lifelong disabilities for many of them. Even if we were to look for it and measure it, there's really very little we can do about it at the moment.
And also, it's a severe threat in immunocompromised individuals, which includes HIV patients, transplant patients, both solid organ as well as stem cell transplant population.
The US Institute of Medicine made it very clear a number of years ago that the CMV is the #1 unmet vaccine need. So clearly everybody agrees this is a problem that needs to be solved. We're committed to do that, and we believe we have the tools to do that. As I said, there are 20,000 children born every year with this infection just in the US, and 6,000 to 7,000 of those every year will have severe disabilities. Just imagine if we can produce a vaccine to prevent that. CMV enters the cell really through two cell types. It enters through what are called fibroblasts and enters through what are called epithelial cells. The key to get into fibroblasts is a GB protein, and that's quite easy to make. The key to open up the epithelial cell is a pentameric protein. It's a protein made up of 5 different proteins that must assemble in exactly the right way.
We were simply able to design mRNAs encoding for the 5 different subunits of the pentamer and co-deliver them into a cell. And the cell's machinery just took over.
What we found and what this study shows is that if you insert the mRNA for those 5 proteins into the body processes them exactly as it would if it had been infected by the virus. And it expresses that protein beautifully, induces an immune response beautifully, and our expectation, and what we saw in animals, is that that immune response could protect against CMV infection. What this data tells us is not only that we have a really good shot at CMV, that it also means that this platform is likely to give us a good shot at any other complex vaccines. And not only single diseases that require multiple proteins and multiple mRNAs, but maybe even single vaccines to protect against multiple different diseases with a single injection.